What I do want to go over is this organelle of the chloroplast, because I don't think I've explained it really in enough depth for this external, because you didn't need to know it super in depth for the internal, but we know that the chloroplast is an organelle. And I like it when that's thick. So we know it's an organelle, the chloroplast, and we know that it is the site of photosynthesis, right? Photosynthesis. And that's where all the magic happens. That's where all the glucose is made. And if we zoomed into a chloroplast, it would kind of look like this. And don't worry about notes or anything today. That's going to be Monday next week's task, to like get all the notes and bits and pieces from this. Today I just want to like teach. So here's our outline, and what the chloroplast has got is got this outer structure here, which is literally just called the outer membrane. So you know how cells have got a cell membrane? Well, organelles have got membranes too. So the chloroplast has got a membrane, and then it's also got an inner membrane. Hence why I drew 2 lines. Inner membrane. And the membrane gives the organelle that structural support, but it also helps to control what comes into the organelle and what goes out. So coming in, we're definitely going to need lots of water coming in. We're definitely going to need lots of carbon dioxide going in, because those things are needed for that reaction of photosynthesis. And then coming out of the organelle, eventually, we're going to have oxygen gas coming out, and we're also going to have glucose. So we're really zooming in now and looking at where this all actually happens. Now, inside this chloroplast, there are these like green pancake looking things. And there are heaps of them. I'm just going to obviously keep it simple and just draw a few. These green stacks of pancakes, and just color a few in. So one of these pancakes is called a thylakoid. So this is a new word that you definitely might not have heard of before, a thylakoid. And if I'll just draw one down here. And this is where all of the chlorophyll is concentrated. This is why they are so green, and this is what makes the plant green. It's all of these thylakoids. This is where all the chlorophyll is concentrated. And so what happens is the sun beams down, and it's the when those photons from the sun hit that thylakoid, it excites the chlorophyll, and that's what triggers this reaction eventually of photosynthesis. So thylakoids are very important. And one more term that I'm going to teach you is a stack of these thylakoids. So a whole stack of them is called a granum, which is just a stack of thylakoids. And what this, the reason why the organelle or the chloroplast is structured like this is because if we can stack as many thylakoids on top of each other as possible, it is going to increase the surface area of these thylakoids so that loads and loads and loads of reactions can happen all at once. Remember, the plant wants to maximize photosynthesis. So the more chlorophyll that can be excited by the sun, the better. So it's got these stacks, and there's like loads and loads of them. And that is what is going to maximize photosynthesis. So Oh, I'll just add one more thing. So in the background here, just this like empty space, this is kind of like, it's kind of like the cytoplasm. So you know how the cell has got all the organelles dotted inside, and then there's this like jelly-like kind of substance that they're all held in, and it's called the cytoplasm. Well, the chloroplast has got a similar type of thing, and instead of being called the cytoplasm, it is called the stroma. These terms are all going to come up again next year when you do level 2 bio, so I think it's good for you to be introduced to it now. So all of that jelly-like backgroundy stuff is called the stroma, and a few reactions happen there too. So that's the organelle. Like I said, don't copy the notes now, that's going to be next week, but the organelle is obviously very important for photosynthesis. And so if I go back to our slides, I wanted you just to recap what photosynthesis is. And so it is this process where plants make their own food, not energy, remember. And you guys already know this equation, but I do want to bring your attention back into what is that glucose actually used for? So the plant makes its glucose, yep, that's fine, but what is the glucose used for? And so I've got these little bullet points down here at the bottom of this slide, and I want you to remember 3 words. If this ever came up in an exam, you would remember these 3 things: storage, growth, and reproduction. Those 3 things, you have to mention them in an exam question if you get asked what, well, in fact, if you get asked any kind of photosynthesis question, because this is the point of it, so that the plant can either store the glucose for later, the plant can use that glucose to make energy, which is needed for growth, or the plant uses that glucose to make energy for reproduction. And then there's a couple of other things, like I've said down at the very last bullet point, to make fat and amino acids and things like that. But storage, growth, and reproduction are the 3 main reasons why the plant makes glucose. And it's the same in humans as well. Like, we have to eat our glucose, of course, but we use it for the same thing. We store it as fat so that we don't like die of starvation if we can't get access to food. We also use the glucose we eat to grow, and we also use that glucose to reproduce, to make our gametes. So just the same in plants as it is in animals. So you've recapped the photosynthesis, and there's a slide here about the chloroplast where I've like drawn a diagram of the chloroplast there for you to copy. And now, to finish off this kind of like photosynthesis stuff, we look at factors that affect the rate of photosynthesis. And again, this is going to be important because I have seen questions like this come up in the past. Again, no need for notes today. I'm just going to introduce it. So there are a load of really cool experiments that we could do, and unfortunately, I don't think we can do any of them at home. So what I'm thinking is that we're just going to do all the content now, and then if we do get back to school, like the week after next, maybe, I don't know if we are, but we're hearing from Jacinda today, we could just do all the practicals like in every lesson kind of thing. So it would just be like practicals for a week even. So that's why I'm just going to do all the content now. And I'm actually going to insert the video of me explaining it right now into this slide so that you've always got that information in this slide. Now, let me get my note back. Okay, factors that affect photosynthesis. Affect photosynthesis. And we've already done one. What were we looking at in the algae balls experiment? I want someone to answer me. Take your mind back to the algae balls. Which factor were we testing to see if it affected photosynthesis?
Light?
Yeah, nice, Maya. So light is definitely going to be one. Another one is going to be temperature. Another one is going to be CO2. So if we do light real quick, because that's one that you already know, I always draw these as a graph, and next year in level 2 bio you're going to have to draw them in a graph too, so I figure I might as well just get you used to it. So if we have a graph like this, and on our x-axis we have got light intensity, and if I labeled it I would just write like low and high. We don't need to worry about like actual numbers. And on our y-axis, which is our dependent variable, we have got rate of photosynthesis. Low and high. And so what we would traditionally see with light is a response like this. And I will explain. So, as you can see in this graph, as you are increasing the intensity of the light, the rate of reaction is going up steadily. And you guys did see that in your results. You saw that as we increased the wattage of those light bulbs, we saw that more oxygen was being produced in those little test tubes. But when we get to about here, we can see that the rate now just plateaus or flattens out. Flattens out / plateau. I can never spell this. E-A-U. Yeah. So the rate has flattened out. It's no longer increasing anymore. And why is this? So the reason why this happens, there's two reasons. The first reason, I'll just put why does the rate flatten. The first reason is because all of the chloroplasts are in use. All of them. So it doesn't matter if you keep increasing light, because it can't possibly create any more glucose or any more oxygen, because all of the chloroplasts are saturated. There's a finite amount. And so to further increase that reaction, we would have to add more chloroplasts. So the chloroplasts are the limiting factor. They are the limiting factor. No, we don't want an S. They are the limiting factor because they are all saturated. Saturated means like full. Saturated. Okay, so that's our first reason why we see this flattening out. And we don't see a decrease in the rate of reaction. It's just steadily happening now at a constant rate. The second reason is because there are other limiting factors affecting the rate. And I think I'm just going to leave that at that point, and I'm going to explain what I mean by that in a little bit. But our other limiting factors are going to be things like temperature, CO2, water, because the plant needs those things too. It could have all of the chloroplasts in the world, but if it doesn't have enough water, if it doesn't have enough carbon dioxide, then that rate of reaction is not going to increase because it needs those things too. So they are called limiting factors. So that's what happens with light. And I'm going to just make it really easy. Carbon dioxide shows the exact same graph. So if we have a graph like this again, but now we've got carbon dioxide concentration, let's say, and then again, rate of photosynthesis, we see a very similar, and low, high, whatever, we see a very similar pattern. So as you increase carbon dioxide, the rate of photosynthesis is going to steadily increase, and it's going to keep increasing, keep increasing, keep increasing until we hit this point here where all of the chloroplasts have become saturated. And so it doesn't matter if you keep increasing carbon dioxide, it's not going to increase the rate of reaction, because all of those chloroplasts are working to the best of their ability. They simply cannot process any more carbon dioxide gas. And so the reasons for the Y are just the same as up here. So carbon dioxide and light intensity are two really easy ones to remember because they're pretty much the same. You just have to change the word between carbon dioxide or light. And also this is very much the same. So there are other limiting factors affecting the rate. And so here with carbon dioxide, you can keep, just say you've got your plants in a greenhouse that's closed off. You can keep pumping more and more and more carbon dioxide into that greenhouse, right? And you would expect the rate of photosynthesis to increase. But if you don't water those plants, or if they're in a dark room and, if you don't provide any light in that greenhouse, they're not going to be able to photosynthesize. So there are other factors playing into this graph, which is why it's a little bit more complex. So this term limiting factors is really important. Okay, last one, temperature. And so plants have got an optimum temperature which they work best at. In animals, like humans, for example, we also have an optimum temperature, but we're lucky because we can regulate our own internal temperature. We've got all these special organs and glands and hormones and things that help us do this. And so our optimum temperature is about 37 degrees. Plants don't have all those things. They are very much restricted to the temperature of the environment that they're in. And plants are going to have a different optimum temperature depending on where they live in the world. So you can imagine that plants that live in a tropical rainforest are going to need a higher temperature to undergo their best version of photosynthesis compared to plants that can be found in the Arctic or something. And because of those evolutionary pressures over thousands of years, they've adapted to the climate in their environment. And so that we prequel, because the optimum temperature is going to be different for every plant, but it pretty much shows a pattern like this. So again, we do it with a graph. In science, we love a good graph, don't we? Graph. And then here we've got rate of photosynthesis. Again, it's always going to be. And on our x-axis, we are going to have temp. Let's just say it's degrees C. Low, high, not going to give specific numbers. And so this graph does look different. Temperature looks different to carbon dioxide and light. And what we tend to see is something like this. As you increase the temperature, the rate of photosynthesis is going to increase. And that is all to do, actually you guys are year 11, so you might not have learned this, but if you think of chemistry and chemical reactions, if you increase the temperature, the particles that are all moving around in the liquid or whatever, they're going to move around faster and faster, and they're going to collide faster, thus speeding up the rate of reaction. And so the same thing happens in plants. And next year you learn all about the little enzymes and molecules and things that actually like control this. But so as you increase the temperature, the rate of photosynthesis is going to increase. And then we hit this point. So this point here is called the optimum temperature. I'm going to write it in red because it's important. Optimum temp. And at this optimum temp, this is where we see the highest rate of photosynthesis. This is where the plant is working at its best. So in the tropical rainforest, that's going to be like 35, 40 degrees. In the Arctic tundra, that's going to be like 5 degrees. Yeah, so it's going to be different for different plants. But the key thing is that this is where it works at its best. Now, once we reach that optimum temperature, it goes downhill very rapidly. So if you kept increasing the temperature, so if you got your Arctic plant and you stuck it in a greenhouse and you made it like 40 degrees, that is going to be too hot for that plant and it's going to die. So what you see is this very, 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 very rapid decrease in the rate of photosynthesis. Rapid decline in rate of photosynthesis. And the reason why you see this rapid decline, just going to explain down here why, is because you get structural damage. So when you add too much heat, you get structural damage to the enzymes, and I'm aware that you might not have heard this word before, enzymes. Structural damage to the enzymes that control photosynthesis. Enzymes are little molecules that are everywhere. We've got them in our bodies too. And without enzymes, we would not be alive. They catalyze all of the chemical reactions in your body. So even when you first take a bite of your toast in the morning, it triggers the production of saliva in your mouth. In your saliva is enzymes. And what those enzymes do is they start breaking down the food in the toast into smaller molecules so that you can digest the food later on. So enzymes are really important, and plants have got them too. And importantly, enzymes control photosynthesis. So all of those chemical reactions to actually do the thing, which you don't look at until next year, enzymes control that. So without enzymes, the plant is going to die, just like we would die if we got too hot. Like a fever. That's why fever is so dangerous in babies, because if those babies get even like 1 or 2 degrees higher than the like 37 degrees that we're meant to be at, it can start to destroy all of the enzymes in that baby's body that is essential for like all of its biological processes. So this is the same, this graph here is the same in humans as well. So those are our 3 factors that can affect photosynthesis. We have got light intensity, which you've already seen with the algae balls. We've got carbon dioxide, which there is a cool practical that we could hopefully do in class later on. And then we've got temperature, and temperature is the one that's a little bit different, because when you increase the temperature too high, you see this rapid decrease in the rate of photosynthesis.